Yes. So please, from now on, the recording is on. If you want to ask a question, make sure you raise your hand so that I know, because then the screen is going to be fully transferred to the WooClap. If you don't want your voice to be recorded, that's your right. But just notice that we already started recording. Okay. So situation. Okay, so we're gonna go through this. Like, I'm gonna give you 1 minute per question. And again, this is not necessarily that the subjects that I asked here are the only ones you have to focus during the exam. This will give you only an indication how the exams will be formulated. So we can go probably there. So that's the WooClap. If you're using your iPhone or your computer, just go to wooclap.com and click on insert the code. If you are in Brightspace, you should be able to see those. And I will start counting. If you want to do it in a setup like the exam, I think you need to go 90 seconds maximum to answer a question. Okay. I'm assuming that you guys have already answered.
We need to access it via Brightspace, and I don't know how to access it on the Brightspace.
Oh, excuse me. So what do you mean you need access on the Brightspace?
So when I went on the web and searched it up, it says that we need to access it through education
Yeah.
platform or
So, but
something.
why you're not using the Brightspace then?
I just don't know where it is. I asked you, but
Where it is. So go
yeah.
under content, and under content you will go all the way down. You will see midterm preparations, and once you click on that, it's inside it. Okay, so probably I would exit this one, and I will get. You see my screen, right, Hayden?
Yeah.
Okay, bear with me here because I'll show you how to get there. So uh, maybe I can share it. Uh, Let other users duplicate. No, not that. So here, copy, and then This one. So now this froze. I don't know why it's not showing me. So we're gonna go to the contents. Oh, it's hidden. Okay, that's what's going on. It's hidden. Now you guys should be able to see it. Maybe it's, I don't know why that. This one, it's visible. It should be visible. I don't know why was that turned off. Okay. Now, are you able to get there? We will know. Okay. Okay. It says. Okay. You got it. Okay. I think that it was this little icon here that was turned off. Okay, let's do it again then. No problems. So we started here, display. Are you seeing my, you still see that, right? So you don't need to share anything else because you should see the screen.
Yeah, we see it.
Okay, awesome. Go ahead. If you want to challenge yourself, answer in 60 seconds. If you want to take your time, it's 90 seconds. Okay. We have 18 people in the group, 8 already answered. We should go ahead and take a decision. Okay, so from next question up, I will start counting the seconds. So I'm gonna stopwatch. Okay. Okay. So what's the correct answer? Recall, we discussed the phosphorylation of the p53 and how is that affected, and the kinase is upstream. So that's the correct answer. Yes, in multiple choices, you always get those noises which will make you think that you are answering correct, like the one here. Inhibition of p53 by the cyclin-dependent kinase p21. P21 is a downstream, it's not an upstream. So p53 affects p21, but p21 doesn't really affect back p53. Okay. You can get those questions and get more rationale, because if we want to do that, probably we can go through all the list of 20 questions today. But as you have the questions, you can Google them and then understand the logic why this was true or false. But if there's something very obvious to be wrong, then we will go through it. Okay. Alexandra, are you answering the question or this is a question I don't get it? Okay, so let's move to the next. Let me take this off and then go to the next. Okay. And I'm counting 1 minute. What happens after p53 phosphorylated activates its target genes? 50 seconds. 1 minute. So what happens when p53 is phosphorylated? Be careful for those who chose it suppresses. Does it suppress apoptosis or it promotes apoptosis? Okay. All of the above. Be careful. Permanently accumulate in the nucleus. No, nothing permanently accumulates. Okay. Let's go to the next question. And counting. One minute. Okay. So which of the following is not? Be careful. That is not a modification. So what are the results? Splicing the introns. One person. Be careful. It's splicing the intron is the most important modification that takes place in RNA, in messenger RNA. Polyadenylation. Yes, it's also very important. There are 3 people who answered no, it's not. So be careful how the question is formulated. It's saying what is not, right? I'm glad nobody put all of the above. And methylation of the promoter has nothing to do with the RNA processing. Okay, so that is the correct answer. Move. Oops, this was answered. Anyway, okay, let's go back to this one. 1 minute. Okay. Is this a true or false? DNA damage, the nucleotide excision repair. It cuts through the 3' and 5' end of the lesion and then replacing it with the right nucleotide. Let me see your answers first. 11 correct. For those who said false, based on what? Um, so it's a nucleotide excision repair. We are removing one nucleotide. So you need to cut through it and replace it with the segment that is correct and seeing the gap with DNA ligase. Correct answer is true. And let's go to the next one. NAD plus is reduced to NADH plus DH by accepting electrons. And in this process, NAD plus is acting as an oxidizing agent. We just discussed this yesterday in the last slides. And 1 minute. Let's see your results. Is this true or false? So NAD plus, does it accept electrons? Yes, it does. And since it steals those electrons as an oxidizing agent for the reduced molecule. So this is a very confusing type of question that you might get. Okay, let's move on. I'll give you like 70 seconds, I would say, for this question. Okay, so what are the answers? Ligands are degraded after use, ensuring that the cell cycle progression. Be careful which ones are fixed and which one are fluctuating. Someone has got the trick. Correct answer is B, because the cyclins are the one that fluctuate, not the cyclin dependent kinases. CDK alone can drive the cell cycle? No. Cyclins bind to CDKs randomly? No. None of the above. So I'm glad most of you got it. Okay. 20 seconds. Okay. Again, reduction and oxidation. What's the results? It's derived from B12, but cannot be reduced. That's the wrong part. NADH acts as a reducing agent by donating electrons to organic molecules. Yes, it donates. It can act as a reducing agent when it acquires those electrons, donating them to NAD plus, NADH, not to organic molecules. Okay. Uh, NADH is an oxidizing agent because it loses electron, like oxidation and losing electron don't go together. So that's the correct answer. Membrane proteins can only be found on the surface of the membrane and they don't span across it. So you can tell the variety of the questions. Some of them are simple, some of them are complex. Could be a little bit more difficult in the exam. So okay. So membrane proteins can only be found on the surface of the membrane. I'll span it. Just a great catch, everybody. We have 16 people. There's one person who is not voting. Is there anyone here who is not really active on the, or you guys, oh, 17, oh, including me probably. You have to try answering the questions because that's how you feel that you are doing something. Okay. 49 seconds. Okay, 1 minute. So what does this proofreading activity of the DNA polymerase do during the replication? Okay. Does it add nucleotides randomly? Of course not. It has to read the template. And it is the one proofreading, not other enzymes doing that. Does not check the base pairing and incorporate. This is totally wrong. DNA polymerase's proofreading function involve adding nucleotide without removing any mismatches. That's totally wrong. None of the above is not correct. So the right answer is B, where it has these three prime nucleus activity. And you can imagine during the exam, I will make those, probably those two options, the option 1 and 3 might be a little bit more confusing for you. Okay. I'm glad you got it. So what happens? What happened during mitosis and meiosis? Both have n chromosome equal 1 or n equal 2. Okay, so still there's like few people didn't answer. In multiple choices in general, you really have to read the question very carefully before you start jumping to the choices. How does the chromosome number in daughter cells compare after mitosis versus meiosis 1? Okay, I can see that this confused you. The answer is probably you directly jumped to answer mitosis versus meiosis, and you didn't realize there's meiosis 1 here. So be careful. That's the correct answer. Because mitosis ends up with 2n, right? And meiosis 1 ends with the haploid ones. Sounds good. Retinoblastoma protein. We discussed this in multiple classes. Okay. So what is the function of retinoblastoma during the cell cycle, and how does it play a role in the regulation of the cell cycle? We discussed this probably during the post-translational modification and the cell cycle progression. Uh, Again, you start to get confused now because we're going back to the classes which were like 3, 4 weeks ago. Does it really promote the transition from the S to G2? Does it really phosphorylate growth factors? Does it really degrade intracellular proteins? Well, obviously there is a three wrong answers. That's why all the above doesn't value. So it blocks the transition between G1 to S phase by holding gene regulatory proteins. And remember that retinoblastoma regulates if the cell cycle has to progress given on the size of the cell. And holding these transcription factor will not allow the cell to pass from the G1 to the S phase because the cell is not in a good size to go through that. But at the same time, I acknowledge that this is one of the difficult questions because it gave you here, it promotes the transition from the S to G2. Remember, P53, retinoblastoma, and P21 are negatively regulating the cell cycle, so it should not promote the transition. It plays a role in opposite way. And then it phosphorylates growth factors. Growth factors are normally secreted outside of the cell. It's not transcription factors, so be careful what type of proteins you are looking about here. Okay, great. Let's move on. Well, what is the last one here? It degrades intracellular signaling protein to prevent cell division. It doesn't degrade them. It just holds them, depends on its phosphorylation state, either bind or not binding. Degradation is something performed by proteasome. Okay. What about the cell membrane? Okay, so as expected, the wording can make some confusion, but think about it. Lipids, they don't like water, right? So if the head is hydrophobic, it has to get, uh, oh wait, I think the question was not formulated correctly. Okay, great. Well, you answered it well. Yeah. The answer was not formulated correctly. I'm sorry. It was like I wanted to confuse you more, but it was like made in a wrong setup. So the hydrophilic head and the hydrophobic tails. I just want to remind you that most of the time I give you these diagrams or figures. Those are really, really helpful when you are studying for your exams. Like for example, here, I'm not sure if I'm sharing the screen. Maybe I have to.
I
Uh,
think your screen is shared, but it seems to be frozen.
I see. Okay, so for example, the hydrophilic head of the, okay, let me just make sure that it's sharing that window. Module, which module was that? Module one. Okay. Ah, yes, here. Sure. So it helps you like when you're studying, the major reason why I add those uh, images is to have a visual memory. So when you're studying and reading that text, it's very easy for you to remember the stuff. Like you can see hydrophilic heads and hydrophobic tails. And also here it's the same. So take advantage of those diagrams when you're studying that while reading, and I get this question a lot from students. I'm not sure how I will study. While you're reading the text, yes, you are speaking about specific molecules and which functional groups, and then you got into a text. If you try to memorize the text, it's going to be super difficult for you. But if you're reading this sentence here and you directly go there and look, then I'm sure you will never forget this. It's a polar hydrophilic head, and that's why it is water from outside, and from the inner side part of the cell, it's also full of water. So that's why the head has to be closer to the hydrophilicity with the water. And the hydrophobic tails, they don't like the water, they are escaping. So that's why they um, form together. So I think, yes, the question, I wanted it to be more complex than that, but you guys skipped it because the first option was more confusing, supposedly. Anyways, uh, take advantage of these diagrams, the bottom line to say that. Okay, so we go back to the questions. Post-translation modifications. Okay, so you guys are getting slow. Are you tired already? So, anyways, so post-translation modification tag the protein by phosphorylation for degradation. Phosphorylation is not meant for degradation. That's ubiquitination. It initiates transcriptional activation by directly binding to DNA, enhancing RNA polymerase activity. Not really. These are exclusively in RNA processing events. Not true. Okay. We're gonna move a little bit faster because we still have few questions. And we have only 10 minutes. I have to move to the next meeting, like right after. So please take a quick look and try to answer. So what defines the primary structure of a protein? Expecting you, but you need to answer this in like 10 seconds, in fact. This is one of the simplest questions. Uh-oh. For those who were hesitating, and when they see the answer of most of the people is number one, please make sure you don't do the same during the exam. There's still some people not answering. But anyways, so bottom line is yes, it's a specific sequence. It's not the folding of the peptide into alpha and beta sheets that determines the primary structures. These are the secondary structures. And multiple peptides is not the first or secondary structure. Addition of a phosphate after translation is not a primary structure. Okay. Which of those is an integral part of an amino acid? Okay. Uh-oh, be careful. Amino acids. Phosphate, sugar, and nitrogen base. This is what? This is a DNA. That's a lipid. Okay. Which part of the amino acid structure determines the unique property of an amino acid? Amino group, carboxyl group, the R group, the central carbon, or all of them. Okay. No, not all of the above. What determines the unique property of each amino acid. What makes one amino acid is different from another? All of them, they have that, and that, and that. It's this difference. Okay. How would an amino acid behave at the pH 7.4 if it's a positively charged amino acid? Be careful that pH can change how an amino acid behave. Wait. So at physiological pH, sorry, at physiological pH, polar positively charged amino acid should be what? Protonated side chain, non-reactive, hydrophobic, hydrophilic, none of the above. So this is basic biochemistry. Please review back the slides because this is mentioned very well. If you have a positively charged amino acid at this physiological pH, it's a positive charge. So it's ready to acquire. Okay. What of the following characteristics is true about non-polar hydrophobic amino acids? So you remember we categorized them into 3 or 4 groups. What is true about the non-polar hydrophobic? That was good. Let's stop with this one. What did you guys answer? They contain ionizable side chain. Ionizable side chain? No. Attracted by water? No. And they tend to be outside of the protein? No. Answer 4. And then you're gonna quickly answer these 2 questions. We have 30 seconds on each. Okay. So what do you think? Oops. What do you think? Okay, we see the confusion is getting high. Okay. I'm not gonna answer this question because it's written in the slides. That you can find it in the slides. Okay. Is there any more questions? No. There's one question probably we didn't cover. It was, yeah. Did you answer it? Yes. Well, one question that we didn't answer. I think it's this one, right? Oh, the one before. This one. Okay, this one. Could you please quickly answer? I have another meeting and I will move on soon. Okay. Uh-oh. You were tricked. Okay. So with this, we're gonna stop the recording.